Welcome everyone. My name is Amy Jark. I'm the Executive Assistant for DPhil, and I want to thank you for joining our webinar today. The program will begin momentarily, but first I want to walk you through just a few housekeeping items. First of all, the participants will be muted during the presentation, which will allow for a clear audio experience. And at the end, we will have a question and answer session. If you do have a question at any time during the webinar, just type it into the question box. And during the question and answer session, I'll read the questions to Dr. Calder and Dr. Blocker. If you do have any just general questions, you just can simply type them into the question box and I'll respond privately to you. Currently, there is a link to the handouts on the home page of the DPhil website. And for those of you who attend the live webinar today, CPE certificate will be emailed to you. If you'd like to view the webinar in the future, we will have it also available on the DPhil website. So today, we'll be doing the webinar. It's entitled The Importance of Nutrition to Healthy Immune Function. And it's given by Dr. Philip Calder. We're thrilled to have him today as he is one of the leading nutritional immunologists in the world. He is currently a professor of nutritional immunology serving on the Faculty of Medicine at the University of Southampton in the United Kingdom. Dr. Felder is a fellow of the Association for Nutrition, a fellow of the Society of Biology, and a registered nutritionist with the Nutrition Society. He has a PhD in biochemistry from the University of Oxford in the UK, a PhD in biochemistry from the University of Auckland in New Zealand, and earned his undergraduate degree in biochemistry from Massey University in New Zealand. He is a member of the UK Nutrition Society, the UK Biochemical Society, the British Society for Immunology, and the European Society for Clinical Nutrition and Metabolism. He has taught extensively at the university level and has received numerous distinguished awards and honors in his areas of expertise. Dr. Calder is a prolific author and has more than 410 scientific publications to his name, and his works have been cited more than 12,000 times. Needless to say, we are honored to have Dr. Calder join us today to illuminate the importance of nutrition to healthy immune function. We look forward to his perspective on enhancing immune function and some of the novel dietary immune interventions. Dr. Calder will begin with a refresher on the immune system, and we'll look forward to a Q&A session after his remarks. So, Dr. Calder, it's all yours, and if you want to show your screen, we can start with the slides. Thank you.
Okay, great. Thank you very much. So, good morning or good afternoon, depending upon where you are. Thanks very much for joining what I hope will be an informative session looking at the interaction between nutrition and the immune system. This has been an area of research interest to me for quite a long period of time. My own specialization is in the area of fatty acid nutrition, inflammation and immunity, but I've also researched on some other aspects of nutritional immunology and of course follow that entire field very closely. So what I'm going to do is start with an overview of the immune system just so that everybody understands the components of the immune system and how it works. Go on to briefly talk about how we can measure immune function in humans. Paint a picture of why getting nutrition right is important for supporting the immune system to operate functionally, and then give you just a little bit of the evidence that shows that nutrition is very important for immune function. And after talking about some classic nutrients, to talk about some non-nutrient but food type compounds that have an effect on the immune system. So let's start with the immune system. What is it? So it's a cell and tissue system that protects the individual from invading pathogens, that is bacteria, viruses, fungi, and parasites. There are many different components of the immune system, and we'll be talking about some of those as we go along. In general, they can be divided into the innate or natural part of the immune system and the adaptive or acquired part of the immune system. The adaptive immune system is slower to respond than the innate immune system, but it's much more specific, and it's specific for particular structures found on bacteria, viruses, and so on, called antigens. So the adaptive immune response is antigen specific. It also has a component of memory, so the immune system can remember its previous encounters with bacteria and viruses and respond more quickly and more vigorously if those pathogens are re-encountered. The components of the adaptive immune system include antigen-specific antibodies and a range of different cells, B lymphocytes or B cells, and different types of T lymphocytes or T cells. These have specific roles, as we'll be seeing shortly. There are also dendritic cells whose main function is to present antigen to helper T lymphocytes. So there's an interaction between these different sites, or these different cells. These cells are also communicating with one another through the production of chemical mediators, the main ones being cytokines. Cytokines are hormone-like proteins that act within the immune system. The innate immune response is actually the first line of defense and is active in the period before the adaptive immune response comes into action. In contrast to the adaptive immune response, the innate response is non-specific. So it doesn't respond actually to a particular antigen, rather it responds to some general structural features found on the surface of bacteria in particular. Because it's not specific, it can act very rapidly, almost instantaneously for some aspects. Unfortunately, there's no memory component to the innate immune response. So previous encounters don't encourage a better response if the pathogen comes along again. The components of innate immunity include another type of protein found in the bloodstream and in other secretions called complement, and some of the white blood cells, particularly the macrophages, the neutrophils, and the natural killer cells. But again, these cells interact with one another through the production of different chemical mediators. So the way the immune system works in general is to distinguish things that belong from things that don't belong. The immunologists long ago called this distinguishing self, things that belong, from non-self, although it turns out this is a rather too rigorous description. And the reason for that is that every day we are exposed to non-self. So we eat animal and plant materials, getting this continuous chronic exposure to non-self antigens of plant and animal and indeed bacterial origin. Excuse me. Of course, it wouldn't be right that we mount an active immune response to all of these non-self components that we come across. And indeed, if we did that, we would have some sort of disease. It might be celiac disease, it might be a food allergy or something like that. So the immune response is also important in recognizing non-self that isn't threatening. So for example, the things we eat. In addition, there are many environmental challenges, including those from bacteria and viruses, that we encounter that actually aren't harmful to us at all. And the immune system is important in distinguishing the ones that are harmful from the ones that are not harmful. So a way to extend or expand this idea of self and non-self is to think that the immune system recognizes and tolerates self, that is our own cells and tissues, and also recognizes and tolerates non-threatening non-self. For example, antigens that come from animal and plant material that we eat. Importantly, of course, it recognizes and attempts to eliminate threatening non-self, that is, the sources of threatening antigens from pathogens. So it doesn't tolerate those things. So the immune response is the person's response to the presence of pathogenic organisms, but it also includes this idea of tolerating non-threatening environmental exposures. It's also involved in protection against cancer cells, and as I've said, it distinguishes and tolerates self and doesn't tolerate non-self. It's typified by interactions between the different cellular components of the immune system, and I'll be talking about some of those cellular components as we go along, and the movement of cells around the body through the lymph and through the bloodstream to sites where infectious organisms are found. Obviously, the immune system is protective to us and highly beneficial to health. Without it, we would suffer infections and probably die as a result. As we've already said, the immune response includes the innate or natural component and the specific acquired component that I've already talked about. I already mentioned it has a component of memory, and that involves the interactions between different types of cells which all have specific roles within the immune response. Some of those cells are to do with processing and presenting antigens. Some of them are to do with communication among the different cells of the immune system, and some of them are actually to do with destroying the source of the antigen, as we'll see as we go along. So how does the immune system work? So I'm going to talk about the four general features of the immune system. The first is that it acts as a barrier to the entry of invaders. So we can think about the skin, we can think about mucosal surfaces of the gastrointestinal, respiratory, and genitourinary tracts, which all keep bacteria and other pathogens out of the body. We can think of the acid pH of the stomach, which kills some bacteria, and we can also think about the presence of harmless, what I've called friendly, bacteria on the skin, within the gastrointestinal tract and elsewhere, which compete out pathogenic bacteria. So in these aspects, the immune system is acting as a barrier to the entry of invaders actually into the body. Of course, there are circumstances where those barriers break down and invaders do get into the body. So the second aspect of the immune response is the recognition and trapping of the invaders. So there are proteins in the bloodstream and in secretions like saliva and tears and so on, and these proteins include complement and antibodies that recognize foreign cells. The proteins coat those cells and enable the recognition by cells of the immune system to be more efficient. So there's a recognition and a trapping aspect. The third aspect is the destruction. So the cells of the immune system are able to distinguish between things that belong and things that don't belong. And the cells that are involved in this discrimination include neutrophils, monocytes, macrophages, T lymphocytes, and B lymphocytes, although they're all working in slightly different ways. These cells can act to destroy what I've called non-self, the invaders that are threatening. There are two general mechanisms to this destruction. The first is a process called phagocytosis. This is the engulfing of the pathogen and its subsequent digestion intracellularly within the phagocytic cell. The second is the destruction of invading pathogens by bombarding them with toxic chemicals released from cells of the immune system. So on this slide, I show those two different aspects of destruction of the invader. So on the left-hand side, we see a phagocytic cell. This is a macrophage, and this is a bacterium. And the macrophage membrane looks like it's reaching out to the bacterium, and then it will actually engulf that bacterium and bring it inside the phagocyte where it's destroyed by proteolytic enzymes and some other factors. So this is a physical destruction of the invader. The second aspect is shown here. This is a cytotoxic T lymphocyte. It could be a natural killer cell or some other types of destructive cells. And this is a virally infected cell. So this is a host cell which has become infected with virus. That is recognized by the immune interaction between these two cells, and as a result of that, proteins are produced from the immune cell, and those proteins lodge within the membrane of the target cell. And they form a particular structure there that actually creates a pore within the membrane, so a hole within the membrane. And through that hole, other chemicals can make their way into the target cell and cause that cell to destroy itself through the process of apoptosis. So we see these two different features, the engulfing and digesting through phagocytosis, and the identification and destruction through the release of toxic chemicals. The fourth and final aspect of the immune response, which I've already mentioned a couple of times, is the memory component. So after an immune response is mounted, some memory cells are formed, and these cells remain in the body for many, many years. Some of them, in fact, remain throughout our whole life. So you all have memory immune cells within your bloodstream that actually can remember the immunological encounters that you had when you were children. What this means is if you get reinfection, you can have a much quicker and more vigorous response. And of course, this is the basis of vaccination, where a small amount of antigen is introduced to a person. They mount an immune response, which goes away, but leaves the memory cells behind. And then if that pathogen, perhaps influenza virus, for example, is encountered, there's a much better immune response because you already have a memory of the previous encounter. So if you like, the immune system has been educated in the way that it should respond when that pathogen comes along. So these four key activities are the exclusion barrier, the identification of non-self and tolerance of self, the elimination, which of course is vitally important, and the memory. So I've mentioned already several different types of immune cells, and this is a very simple cartoon showing the interactions amongst some of those cells. So because time is tight, I'm just going to talk about the left-hand side here, but that is sufficient to illustrate my point. So here we have some bacteria which are identified through the innate or natural immune response, including the phagocytic cells. The phagocytic cells engulf the bacteria and destroy it. Some of the components of the bacteria, the antigens, make their way to the surface of the phagocyte, and once on the surface of the phagocyte, they can be displayed or presented to a T helper cell, which actually is predetermined to be specific for that particular antigen. Once the T helper cell encounters its antigen, it becomes activated. When it's activated, it will release these hormones called cytokines that I already mentioned. And the role of the cytokines is to increase and regulate the activity of other cells within the immune system. So, for example, some of the cytokines released from the T helper cell will act on B lymphocytes, making the B lymphocytes produce antibodies that are specific for the original antigen. Those antibodies coat the bacteria and make the process of phagocytosis much more efficient. So with this simple example, I've shown you the interaction between three different types of host immune cell that act to make the response to this particular bacterium much more efficient. Of course, the immune response is much more complex than that, and this drawing is simply to show you that there are many, many different types of immune cell other than what I showed you on the previous cartoon. There are many different types of interactions directly between those cells, and there are many different chemical mediators that are produced interacting amongst the cells. The important thing to note about this apparently complicated figure that we're looking at now is that it's been called the basic immune response from a second-year medical student's lecture notes. So you can imagine that the reality is much more complex than this. The other thing is that this diagram was drawn in 1996. So that's 13 years ago. And again, you can imagine the immunologists have been very busy discovering new types of cells, new subsets of cells, and new chemical mediators. So the real immune response is much more complex than even this diagram that we're looking at here. And of course, that creates a challenge if we want to understand the impact of nutrition within the immune response. One way to think about how these cells are interacting with one another is perhaps to use an analogy with an orchestra. So we have the different instruments in the orchestra, which can all, of course, make their own music, But the sound of the orchestra is much better when all of the different instruments are playing in an integrated and coordinated way, each playing their own individual roles, but making a contribution to a much greater whole. And this response of the orchestra is coordinated by the conductor in the middle here. And in the immune response, we would call the conductor the T helper lymphocyte, as I showed you earlier on, coordinating the action of all of the different types of immune cells, mainly through the production of different chemicals, particularly the cytokines. So the cells of the immune system, of course, are found in the bloodstream, they're found in the lymph. They all originate in the bone marrow. Some of them mature in the bone marrow, and some of them mature in the thymus. But once mature, those immune cells make their way throughout the body to different organized secondary lymphoid organs. These include the spleen, different lymph nodes, the tonsils, and the gut-associated immune system, which I'm going to talk about later on, but also organized immune compartments in other mucosal surfaces, for example, in the respiratory tract. So the point is that the immune system is dispersed throughout the body because, of course, exposure to pathogens can occur anywhere in the body. It's obvious that having an immune system that works properly is important in preventing us from being infected, and also from controlling, in controlling what happens once we are infected. What about the life course? So when a baby is born, it has all of its immune complement that it needs. It has all of the different cell types, but they don't really know what to do yet. So the immune system is functionally immature. It develops from soon after birth, partly through the exposure to maturation factors in breast milk, and partly through the exposure to new antigens found, for example, on the mother's skin, in the environment, and once foods are introduced, through those foods, and through exposure to pathogens in the environment. So this period of immune maturation occurs over the first few years of life, and of course is vitally important. At the other end of the life cycle, our immune system declines with age. This is a process that's been called immunosenescence. And basically it seems that our complement of immune cells becomes used up as we go through the life cycle. So we get more and more memory cells, and we have fewer and fewer cells that haven't encountered antigen. These are called naive cells. So our immune competence declines with age, particularly if we become frail. And what this means is that in both the newborn and in elderly persons, there's an increased susceptibility to infection and an impaired ability to deal with pathogens. So these are very vulnerable times of the life cycle. So if we're interested in nutrition or aging or exercise and the immune system, we have to be able to measure immune function. So how are we going to do that? Well, there are quite a different number of approaches that can be used. So in the simplest case, we could simply take blood or other accessible sites like saliva and measure immune markers in the blood. We can measure cells, we can measure antibodies, we can measure complement proteins, and so on. So if you like, this is a measurement of immune status in a particular body compartment. We can also isolate immune cells, particularly from the blood of humans, and study how those cells respond when they're placed in culture. And typically what we would do is we would put the cells in culture and then expose them to some sort of immunological stimulus and see how they respond. And it could be how do they produce a particular cytokine, it could be how well do they divide, can they produce a particular type of antibody, and so on. So these are ex vivo measurements of immune cell function. It's interesting to note that if we make those sorts of measurements, there's quite a variation amongst different individuals. So this is data from one of our own studies using cells taken from the blood of healthy middle-aged men, and looking at the function of those cells in culture, including this function of phagocytosis that I talked about before, ability to produce superoxide radicals, which are damaging to pathogens, and the ability to produce certain cytokines. And the key thing for you to see here is simply there's a lot of variation among different people. So this, of course, is because those people are different in many characteristics. They might have had different pathogenic exposures during their life course and so on. So immune responses differ among different people. The final ways to study the immune system are to actually study what happens within the body in response to an immunological challenge. So experimentally, we could give people an immune challenge like a vaccination, and we can study how they respond to that vaccination. So this is an increase in anti-vaccine antibodies in people who were given a vaccination at this time point here. So we could study this in the context of a nutritional intervention or some other sort of intervention, or to look among different subgroups of the population. Finally, of course, we can look at the pattern of infections. In humans, this is a little bit difficult because normally we're reliant upon natural exposure to infectious agents, but we can ask questions about, do infections occur, how severe are they, how long do they last, and so on. So there's a whole range of different approaches we can use to study the immune system. Let's move on now to nutrition. So why is nutrition important to supporting immune responses? So what I'm showing here is that in a normal situation, let's say, the immune system is at rest. Of course, it's undergoing its normal surveillance activities, but it's not particularly active. Once it encounters a pathogen, it becomes highly active, and there's an increased demand for energy and for building blocks, for the production of new cells, for the production of all these different chemical mediators that I've been talking about. The building blocks for that have to come from somewhere. Also, some of the substrates for chemicals involved in the immune response actually are nutrients. So, for example, the amino acid arginine is a precursor of nitric oxide, which is very important in antimicrobial defenses. Other nutrients, for example, vitamin A, vitamin D, and zinc, are regulators of the immune response. So we have energy sources, we have building blocks, we have substrates for the synthesis of immune factors, and we have regulators of the immune response. And these are all important roles for nutrients. So there's a demand by the immune system for a whole range of nutrients, and of course this demand can be met from the diet or from body stores. In turn, of course, the body stores have come from the diet. So ultimately, nutrition is the source of the things that the immune system is demanding for it to behave optimally. So now I just want to show you some of the evidence that links nutrition, nutritional state, and specific nutrients to immune function. So let's start with malnutrition, by which I'm really meaning undernutrition. So it's long been known that undernutrition impairs immune function and increases susceptibility to infection, and that these impairments can be reversed by improving people's nutritional status. So this is a rather old study now, but it makes the very important point that I'm telling you. So this is a study in malnourished children, or children with varying degrees of malnutrition, who received an immune challenge to the skin, and what the study showed was the more malnourished the children were, the poorer their immune response was. Of course, the potential for improved nutrition to save lives through improving host defenses among the malnourished is enormous. So around about half of global deaths are due to undernutrition, and half or more of those are due to infectious agents. So you could imagine, roughly speaking, perhaps a quarter of global mortality is due to immune impairments as a result of undernutrition. So this is millions of lives that could be saved through better nutrition, particularly in developing countries. Let's look now at specific micronutrient deficiencies. Of course, a whole range of vitamins and minerals have been studied. Specific micronutrient deficiencies impair immune function, increasing susceptibility to infection. And again, these impairments can be reversed by correcting the deficiency. So this is an example of such a study. So this is a study in vitamin A deficient infants in Indonesia who were put into a randomized controlled trial of placebo versus vitamin A, and after a period of weeks they were given an immune challenge, that is, tetanus vaccination. And this is the response to the tetanus vaccination. So you'll see the response was much greater in those children who had been vitamin A deficient and had now been made replete compared with those who were still vitamin A deficient. So far, the examples I've shown you have been from developing countries. But in fact, these effects of poor nutrition are also seen in developed countries. So this is an example of a study from homebound but fairly healthy elderly women studied in Canada. So the women were recruited into a study and a range of immune functions were measured using cells isolated from their blood, and their iron status was measured. Some of these women had low or deficient iron status, and some of them were sufficient. And it turned out that immune functions were all impaired in the iron deficient women compared with the iron sufficient women, as shown here with one particular example, simply showing that this is T lymphocytes from the blood of the iron sufficient women responded much better in a test tube than those from the iron deficient women. So these sorts of studies have been used to show that a whole range of nutrients are very important to supporting the immune response in humans. These include various amino acids, the sulfur-containing amino acids, glutamine, arginine, the branched chain amino acids, particularly leucine, some amino acid metabolites, and the special amino acid taurine are all important in supporting immune function. Essential fatty acids and omega-3 polyunsaturated fatty acids are very important. The fat-soluble vitamins, the carotenoids, the B vitamins, folate, vitamin C are all important. Probably all minerals are important to supporting the immune response, but zinc, copper, selenium, and iron are amongst those that have been investigated most widely. And some non-nutrients that are found in the diet also turn out to be rather important, including some of the plant polyphenolic, flavonoid-type compounds, and also some non-digestible carbohydrates found in some vegetable materials, so the prebiotic-type saccharides. So the idea that I've come to, that perhaps I've convinced you of, is that getting nutrition right, so what I've called here an appropriate nutrient supply, means we have a good nutrient status and good nutrient stores, and that means the immune system can function appropriately when it's challenged. Now you could replace the words appropriate, I guess, with optimal here, although it's a little bit difficult to define exactly what optimal immune function means. So I've used the word appropriate. But the key idea is simply that getting nutrition right is very important to support the immune system in its roles. And the extension of that, of course, is we're not just actually interested in immune function, we're interested in the ability to defend against pathogens and respond appropriately to other immune challenges. So again, getting the nutrition right gets immune function right, which means we have better host defenses. So an example of that is shown here with vitamin D and the flu. So this is a fairly recent study in Japanese school children who were taken into a randomized controlled trial of placebo versus vitamin D daily for 16 weeks during the flu season. Quite a large study for a human nutritional immunology study, about 320 children studied in total, and the outcome was, did they get the flu or not? So in the placebo group, almost 20% of the children got the flu. In the vitamin D group, the number was about 50% of that. So there's a reduction in infections in the study, which must be due to something to do with the impact of vitamin D on the immune system, enhancing host defense against the influenza virus. So I just want to divert slightly into another couple of areas of nutritional immunology which are quite interesting. So these two slides that I'm going to show you now summarize information in a review written by Gabriel Fernandez from Texas, who has been a pioneer of the study of caloric restriction in animal models. And what Fernandez and others, of course, have shown is that caloric restriction in rats and mice prolongs lifespan, decreases the development of age-related chronic disease, and has an impact on antioxidant defenses, and also improves immune function. So there's something about caloric restriction that has an immune effect. Of course, this isn't creating a nutrient deficiency state, it's simply reducing the amount of energy. And there's a good provision of all of the essential nutrients and so on in these studies. What Fernandez went on to show was that the combination of caloric restriction and omega-3 fatty acids from fish oil actually improves the immunological situation compared with caloric restriction alone. So there is a decrease in inflammation, which causes some of the age-related chronic diseases, but also an increase in immune competence with the combination of caloric restriction and omega-3 fatty acids. So the big question from all of these studies of nutritional immunology is, can we improve people's resistance to infection through improving immune function? Now, I've already given you a hint that the answer to that is going to be yes, because I showed you, for example, a trial in Japanese school children given vitamin D. But it's possible that those children had a deficiency of vitamin D that was corrected. So I think the answer to the question is certainly yes in deficient individuals and malnourished individuals. In other words, if we take deficient individuals and make them replete, or we take malnourished individuals and get them well nourished, we will improve their immune competence and we will decrease their susceptibility to infection. The big question is, what happens in normal, healthy, free-living individuals in the developed world? And here the answer isn't actually quite clear, although there are some situations where the improvement in immune function has been correlated with improved resistance to infection. The last part of my talk is about novel dietary immune interventions. So these are nutritional interventions with things that you wouldn't normally consider to be nutrients. And a lot of these studies have focused on the gastrointestinal tract. And the reason for that is twofold, but one reason is about 70% of the human's immune cells are actually found lining the gastrointestinal tract. And the reason for that perhaps is obvious. So this is a diagram here of the gut epithelium. This is the gut lumen out here shown in blue, and this is the underlying gut wall. So the reason we have a huge immune component within the gastrointestinal tract is twofold. One is, of course, the gut lumen is full of microorganisms, particularly bacteria, and we have to be able to deal with the bacteria that are living in our gut. The second thing is, of course, we have this constant availability of foreign proteins from the animal and plant material that we're ingesting. So we have to have a system to educate the immune system to tolerate these foodborne antigens. So there's two aspects here: the close proximity of bacteria and the presence of proteins of animal and plant origin. So the gut-associated immune system is highly organized, as this diagram shows. I won't go into detail, but the important thing for you to know is there are particular structural entities to this immune system, and there are particular ways that the immune system can link with and identify antigens, be they of bacterial or food origin, within the gut lumen, and they carry those signals back to the secondary lymphoid organs like the lymph nodes and the spleen, where the immune system works out what sort of response it should mount to either the food or to the bacteria. So you can imagine this is very, very important. So people have taken this idea and they've extended it to adopt the hypothesis, if you like, which is now pretty well accepted, that there are specific interactions between bacteria in the gut and the gut-associated immune system by direct interaction between the bacteria and the immune system and through chemical mediators, and therefore the gut bacteria are playing a very important role in educating the host's immune system. So this is an example from a review article simply showing you that people have been working out a range of different mechanisms by which gut bacteria interact with and communicate with the gut epithelium, but also the gut-associated immune system. So there's lots of different interactions going on here. So if this is true, there's a question of whether if we change the gut bacteria, can we impact on the person's immune response? So we could change gut bacteria and impact on host immunity, or we could perhaps provide some compounds which are taken up directly by the gut immune cells. And I just want to give you an example of each of those approaches before finishing. So probiotic bacteria are bacteria that colonize the gastrointestinal tract and have some sort of interaction with the host. So this is an interesting study in healthy elderly persons in a randomized controlled trial who were given a normal yogurt or a yogurt containing particular probiotic bacteria for 13 weeks. Quite a large study, about 200 elderly people in total. After 4 of the 13 weeks of intervention, the subjects received the seasonal influenza vaccination as an immune challenge. You might know that the seasonal flu vaccination includes 3 different virus fragments. So you get an antibody response to each of these virus fragments. So the figure on the right here shows the response to each of those viral fragments in the control group in the open triangles and in the probiotic group in the closed squares. So the idea is the better the response to vaccination, that is the higher the antibody response, the more protected the person will be. And you'll see the response was always higher in those individuals taking the probiotic compared with control, although this was only statistically significant for 1 of the 3 viral strains. Nevertheless, this is a study which shows that altering the composition of gut bacteria has an impact on host immune responses. The second approach has been to directly interact with the host immune system, not through changing gut bacteria, but through an interaction with those gut-associated immune cells. So Wellmune is a beta-glucan from yeast. When taken orally, it actually interacts with these particular structures within the gut-associated immune system that I showed you before, the Peyer's patches. Then the immune cells actually travel from the gut-associated immune system throughout the body, as I indicated they did before. And what this means is the interaction at the level of the gut is actually transmitted or translated, if you like, throughout the body. And what this means is an effect through the gut can actually be manifested systemically with regard to the immune system. And it turns out that this particular beta-glucan makes neutrophils work much more efficiently, particularly with regard to phagocytosis and the intracellular killing of bacteria. So this is a study using Wellmune in medical students looking at cold and flu-like symptoms in a randomized controlled trial setting. So it was a 90-day study in 100 students who were randomized to placebo or 250 milligrams per day of Wellmune, and over the course of the study their cold and flu symptoms were monitored using this validated questionnaire, and there were also some immunological parameters that were measured. The key finding of the study was that the number of days with cold and flu-like symptoms were reduced in those students who were taking the beta-glucan compared with placebo. And this was a reduction of the order of about 30% or so. It didn't quite reach statistical significance, but clearly with a larger study and the same result, this would have become statistically significant. So there's an effect actually of beta-glucans, of this particular yeast beta-glucan, on infection in this study. The last study to show you is a study where exercise was used as an immune challenge. It's a crossover study in 60 people who consumed placebo or Wellmune Beta Glucan for a period of time, and there was an exercise challenge whilst they were on both the placebo and the Wellmune arms of the study, and the outcome was immune responses before exercise, immediately after exercise, and sometime later on. So this is an example of the readout from the study. So remember, this is a crossover trial, so these are the same individuals on the placebo arm and on the Wellmune arm. The measurement here is production of the cytokine interleukin-4, which you see was greater when the subjects were consuming Wellmune, particularly in the period immediately after exercise. So there might be better immune defenses to an exercise challenge and perhaps other immune challenges in individuals taking this yeast beta-glucan compared with not. So my take-home messages are that the immune system is important to a healthy life. It's very complicated, involving many cell types and many chemical mediators. It develops early in life and declines with aging. It's possible to measure many different aspects of the immune response in humans. Poor nutrition is associated with a poor immune response and increases susceptibility to infection. And these can be improved by improving nutrition. There's extremely good evidence that having a good diet is very important to supporting good immune function. And there are some novel dietary interventions, including prebiotics, probiotics, and yeast beta-glucans, that might have specific roles to improve the individual's immune response. Thanks very much for listening. Hopefully there are a few questions now.
Yes, thank you very much, Dr. Calder. We do have some questions from the audience for you and for Dr. John Blocker from Biothera R&D, who will help answer some of those specific questions. I'll start with the first question. How well can the body repair itself after it experiences extreme stress and has been extremely compromised?
Yeah, I think this is an important question, and of course we have quite a capacity to repair our cells and tissues, but there are some situations where, you know, things have happened that are beyond repair. But what's interesting is that nutrition is very important in supporting many aspects of repair, including, you know, the immune component, the wound healing component, and so on. So there are situations, for example, of very severe trauma, of burns, where nutrition is part of the interventions that are used to create a better outcome. So I think, you know, we have a capacity to repair ourselves. Immunity is part of that capacity, and therefore good nutrition will be part of that capacity as well.
Okay, thank you. The next question is from Carmine Mina, and she asks, what is the most accurate way to test for both food allergies and food intolerance, since immune response is included in both?
Yeah, so I think for food allergy, you could, I mean, the gold standard is a double-blind, placebo-controlled food challenge, where you slowly introduce increasing amounts of the food or the food component that you think a person is allergic to. So this would be a gold standard for food allergy. Of course, you would have a clinical history and some other things that would lead you up to using the food challenge, because that, of course, can be dangerous to the individual if they show an anaphylactic reaction. So you need a particular clinical setting to do that sort of thing. A food intolerance is a little bit different, because a food intolerance may not necessarily always be an immunological reaction. So there will be types of a food intolerance, like celiac disease, for example, which is immune-mediated, where you can measure particular specific markers that would be diagnostic for celiac disease in the bloodstream. But there may be other forms of intolerance where you can't do that, and you would be using an elimination and perhaps a reintroduction in a diagnostic sense.
Okay, thank you. The next question asks if you would please discuss the relationship between nutrition and autoimmune diseases or conditions. It's a big topic.
Yeah, I think that in a way that's a topic for another day.
Yeah.
An autoimmune disease is where this idea of tolerance to self has been lost. So your own immune system starts recognizing your own antigens and mounting its destructive activities against places where those antigens are found. For example, in someone with rheumatoid arthritis in the joints, and so on. So all of these diseases have a genetic component. There's a genetic predisposition. But they all also have an environmental component. And often one of the environmental components is an infection. So there's something to do often with a specific type of infection initiating an immune response, which then becomes abnormal. Now, it turns out that nutrition can be important as well. So, for example, vitamin D is believed to be very important in preventing some types of autoimmune reactions. And there are animal models of some autoimmune diseases where you can actually cure those animals with vitamin D. Omega-3 fatty acids have also been used in some of these animal models, and they have been used clinically in some autoimmune diseases like arthritis. Antioxidant vitamins might be important because there's always an element of oxidative stress. So I think there is a role of nutrition in creating an environment where, there's a role of, let's say, poor nutrition or unusual nutrition in creating an environment where autoimmunity can manifest itself. And therefore, again, you might be able to use nutrition to help modulate and bring about some clinical benefit in those diseases. But I think in humans you couldn't really use nutrition to cure those diseases, but you can use it to decrease the severity, I think.
Okay, thank you. The next question is from Amanda. She was asking, are food antigens ever harmful? Many doctors conduct food allergy blood tests and encourage patients to reduce consumption of certain foods due to lab results. Is this advice supported by science, and can it affect immune function?
Yeah, so all food contains antigens, and in a healthy individual, our immune system recognizes those antigens and recognizes that there's nothing to worry about. So if it mounts what's called a tolerogenic response. If it mounts an active immune response to a food antigen, then that person has an immunological allergy to that food. And this will be manifested usually with gastrointestinal symptoms, but it could be also with respiratory and skin symptoms, for example, atopic dermatitis or asthma in response to the food. So this is a genuine immune-mediated disease that can be diagnosed by measuring the combination of the anti-food antigen antibodies in the bloodstream and the use of the food challenge that I talked about before. And that's because there are people who have the antibodies in their bloodstream, but they don't actually mount, or they don't have the disease. And of course there might be some people with the disease who don't have high levels of the antibodies. So I think for a genuine allergy you always need the gold standard food challenge. My understanding is that the professional allergists do not believe strongly in many of the tests that are used by some clinics, by some doctors, and might be available over the internet. So I think if you're a responsible dietitian or health professional, you would always use the most appropriate, most recognized tests that are accepted by the medical community.
Okay, thank you. The next question is, what does Dr. Calder think about mediator release testing for food sensitivities?
Yeah, so this is the sort of thing that I was just talking about. So this would not, I mean, if you have a food allergy, one of the things that's manifested is mast cell degranulation, for example, and that causes hypersensitivity. So this is a key immune component to food allergy. So therefore, people who are food allergic might show in a test tube mast cells and other immune cells that are very responsive. So that might be an indicator, but it's not accepted as a diagnostic test. So again, I would revert to my previous answer that you should, as you would always do, I think, use the most well-validated and well-accepted gold standard tests for a diagnosis. Of course, these tests can be a useful pointer for some people, but I wouldn't use them diagnostically because the professionals do not accept them in a diagnostic sense.
Thank you. Would you comment on the various T helper cells when it comes to autoimmune disease? She says, I'm familiar with Th1 and Th2, but I'm learning that Th16 also has an impact on autoimmune.
Yeah, so the immunologists have been very busy discovering new types of T helper cells. So you're right, you mentioned Th1, Th2, and the old idea was an imbalance between Th1 and Th2 cells will underlies disease. Now this is still true, but there are other T cell subsets. So there are T regulatory cells, which are a little bit like the brake on your car, if you like. So T regulatory cells put a brake on immune activity. So it could be that in some autoimmune diseases you've lost the ability of T regulatory cells to function, so that the immune system is just running away with itself. The other thing is there's another subset of T helper cells, it's Th17 actually, that seem to be involved in autoimmune reactions. And there's an association between people who have autoimmune disease and higher levels of Th17 cells. There are also some other types of T helper cell subsets. There's a Th22 cell, for example, which seems to be involved in asthma. So the questioner is right. There are increasing numbers of T helper cell subsets that are involved in diseases and will, I think, perhaps in future be part of a diagnostic panel of immune markers that can be used. Of course, in none of this is it like an all or none or a black and white. You know, there's a gray area, I think, and this is where research is very important to work out what's going on.
Okay, thanks. The next question, and it might be directed to Dr. John Blocker. Is yeast beta-glucan considered a prebiotic?
It can function as a prebiotic because it is fiber, a source of fiber, but at the level that we've conducted our clinical trials at 250 milligrams per day, it's probably not a significant source of a prebiotic.
Okay. Another question about the beta-glucan. It says, what food sources provide beta-glucan besides the product you mentioned?
Beta-glucan can be a pretty confusing term because it covers a very broad range of food components. There are beta-glucans in cereal grains. That's the ones we're probably most familiar with from oat and barley. Those have no direct immune effect. They, in fact, they're a tolerated antigen, essentially, as Dr. Calder has been talking about. The ones that have the direct interaction with immune cells are going to be from yeast and potentially some fungal sources.
Okay. The next question again, how are the yeast beta-glucans developed or isolated? Are they specific to a certain type of yeast only?
In the case of our product, what we do is we take the cell wall from yeast and we strip off, we basically purify by removing through hot water washing all the other components, so that you're dealing with a material in Wellmune that's 75 to more typically about 80% pure 1316 beta-glucan from yeast. So that's what we're doing with it. The other sources that might be available are typically not going to be that high of beta-glucan content.
Okay, and would someone trying to heal the gut by decreasing candida yeast overgrowth in the gut be able to take and benefit from Wellmune WGP?
And I'll give an answer, and Dr. Calder, if you've got a comment on that. Basically what's going on with Wellmune is the interaction is not so much in the gut level itself, it's taken up by the gut, but you're looking at a systemic immune reaction from the immune tissues throughout the body and in the innate immune system. I'm not sure there'd be a whole lot of interaction with what's going on with gut flora when you take Wellmune.
Yeah, I don't think it's possible to comment specifically on the question because the research hasn't been done. But there are nutritional strategies that have been used to change, let's say, the load of pathogens within the gastrointestinal tract. So this is where some of the and more particularly the probiotics have been very useful, not necessarily with yeast infections, but with particular bacterial infections. This has been quite well documented for some interventions. So, I mean, it's possible that through direct actions in the gut, but also actions through the immune system, you can affect pathogens within the GI tract.
Okay. And is there any advantage to taking Wellmune twice a day?
At this point, all of the 9 human clinical trials that we have conducted with Wellmune have used a once a day in the morning for dosing of the product. Theoretically, you could potentially see a little bit higher uptake if you split the dosing up a couple of times a day, but that's theoretical and not based on the actual clinical evidence.
Okay. And the last question is approaching, it's at 1 o'clock, so the question is, should a person with an autoimmune disorder such as RA avoid all immune boosting supplements?
I can talk specifically with Wellmune, and basically the answer to the question is we don't have any data. With Wellmune, we have conducted, again, the 9 human clinicals we've done have all been with healthy, normal populations, so we do not have data on people specifically with compromised immune systems. Dr. Calder, do you have any comments?
Yeah, I think what you have to keep in mind is that in autoimmunity, the adverse immune reaction is being driven by a specific antigen. So you have an overreaction to that antigen and a disbalanced immune response to the antigen. And I don't really think that, I mean, you use the term immune boosting, which is not really a term I particularly like, but I don't think creating a situation where the immune system might work better is actually going to make autoimmunity worse, if you see what I mean. Because we're talking about nutrition sort of setting the environment within which the immune system can work. We're not really, except for 1 or 2 examples, talking about nutrition having a specific interaction with the host immune response. Of course, you know, I'm talking theoretically. I'm not sure that there have been too many studies done that can address this specific question. I mean, it's an interesting question, I think.
Okay, thank you. I think Before I end, it's after 1, so I just want to thank everyone for joining us today, and I want to remind everyone to check out the DPhil website for the following. There are the webinar handouts that are on the home page of the website. The webinar recording will also be there. It should be up by tomorrow. Also, there were quite a few of unanswered questions today in the Q&A session, and we will provide a link to answers to those questions. So just check the homepage of the website, and there will be a link there. You can link directly to the answers. Also a reminder to sign up for the upcoming webinar on April 11th at 8 o'clock Eastern. It's called Keys to Understanding Clinical Studies on Nutrition and the Immune System, and that will be presented by Don Cox, PhD. He's the Senior Vice President of Biothera R&D. So if you are interested in joining us for that webinar, you may send me an email, and I will send you the active registration link when it's ready. So again, thank you everyone for joining us, and thank you Dr. Calder and Wellmune. I appreciate you putting this webinar on for us today.